I didn't see you. Thank you very much, Nikki. Yeah, no, yeah, thank
No
you very
problem.
much.
Yeah.
I already got
Please.
I already got some answers from people from Zeiss and from other users in general. So yeah, that was very helpful. I'm going to continue exchanging with them.
Fantastic.
Thank you
Feel
so
free to
much.
reach out. Feel free to reach out to us as well.
Definitely. Maybe I can already share my screen. Let's see.
And I can introduce you briefly once everybody settles in and starts writing their names into our, which I'll put in the link to our doc.
Sure.
And everyone will take attendance first, and then after that, I'll give the introduction.
Sure. And whoever is the host, can you let me share my screen?
Ooh.
Nikki, you're a co-host, so you should be able to do that along with Claire.
Yep, let me find. Oh, there you go.
If you guys want to make me a co-host as well,
Sure.
I'm happy to help.
So now you're seeing my the main screen, the first screen of my amazing.
I added you on the attendance, Natalie.
Thank you. So we'll just give it a minute or two and we'll get started at 9:05.
Sure.
All right, so thank you everyone for being here today, and thank you, Mathias, for joining us so early from the West Coast. So Mathias obtained his bachelor degree in life science at the University of Paris-Saclay and his master's degree in neuroscience at Sorbonne University. In parallel, he attended the Ecole Normale Superieure Paris-Saclay in France, where he received a master's degree, and he recently joined, I guess not so recently, but he joined Anne-Marie Craig's lab in 2019 for a year-long internship where he started to work on the magnified analysis of the proteome, so MAP, for the study of excitatory synaptic proteins. He came back to this lab at the end of his master's to start a PhD with Anne-Marie Craig and Mark Cembrowski in May 2021 and finished the work of the adaptation and characterization of MAP. He is now focusing on the study of temporal lobe epilepsy. He plans to use the adapted MAP protocol to study the synaptic pathology of this disease at the protein level, and in parallel using a state-of-the-art single-cell spatial transcriptomic platform called Xenium from 10x Genomic to study the changes of gene expression in temporal lobe epilepsy. So thank you, Mathias, and I'll hand the stage over to you.
Thank you very much, Natalie, and thank you very much for the invitation to give a talk today. It's really my pleasure to share a bit of my work with you all, and especially with people who are a bit familiar and maybe wanting to use expansion microscopy or just hydrogel-based techniques, because I think they're very promising, but maybe we need a bit more coordinations and discussions between users to fine-tune the different protocol of these techniques. So yeah, as you were talking about, the first part of my PhD was to finalize the adaptation of the magnified analysis of the proteome. This technique was the one that was developed in parallel to the expansion microscopy. It's the same concept as the expansion microscopy, but with a slightly different chemicals used, a slightly different composition of the gel. And so I'm going to show you how we adapted it a tiny bit, and especially how we characterize this technique for the study of excitatory synaptic proteins and how it can help to evaluate the cell type specific distribution of synapses in the brain. So I'm currently also co-supervised by Mark Cembrowski and Mary Craig, as Natalie mentioned. So as we all know, synapses are extremely complex and heterogeneous in their protein composition, with a very high density of proteins within a very small space. There is less than 100 nanometer between the presynaptic active zone and the postsynaptic density, which makes it challenging to visualize and study synaptic proteins directly in preserved brain or brain sections. The gold standard has long been, to study this structure, has long been immunogold electron microscopy. However, volume which can be acquired in a reasonable amount of time is small. While light microscopy, which can cover bigger volume faster, presents other limits, the resolution and limited antigen accessibility, as we need antibodies to access the proteins of interest. So we have been working on the magnified analysis of the proteome to apply and adapt it independently to the study of synaptic proteins. And I think independently is quite important because many of the publications that we've seen in the literature are mainly from the initial labs who developed these techniques, or from postdocs coming from these labs who developed these techniques. So having some people independently applying these techniques are also very important to show the capabilities and the reproducibility of this technique. So I'm not going to extend myself too much about the concept of the expansion microscopy, as you may all know. What it is to make a tissue physically bigger, to add physical magnification to the optical magnification of the microscope. And to do so, the tissue is embedded in a gel of acrylamide, which reacts with the proteins in the tissue. And then after washing the hybrid with detergent, most of the stellar components are removed except for certain proteins. And then incubating the resulting hybrid in diluted PBS allows its isometric expansion in all dimensions. Maybe I can mention here that one of the main difference here between the magnified analysis of the proteome and the expansion microscopy is that the composition of acrylamide is way higher in the magnified analysis of the proteome. And also initially, in the initial protocols that were released, the denaturation or digestion step happened before the staining, the labeling with the antibodies. And so in our case, because we wanted to get this gain of antigen accessibility, we found it better to do the labeling after the digestion, after the denaturation, in order to have an increased antigen accessibility, while in the original expansion microscopy, the antibody labeling was performed before the digestion or denaturation. That's why at the beginning we started to work on the magnified analysis of the proteome rather than expansion microscopy. So this resulted in a recently published paper, Adaptation of Magnified Analysis of the Proteome for Excitatory Synaptic Proteins in Varied Sample and Evaluation of Cell Type Specific Distribution. So you can find this paper, and also associated with these papers, the two main protocol, the step-by-step protocol that we've been adapting a little bit. And this protocol, we let them in on access in OSF, Open Science Framework. And so with the returns I'm going to get from multiple users that may want to use these protocols, I will update or describe better, detail better the protocol, just to make sure that everyone can use this protocol without having external help to apply it. So for this study, we had three main aims. We focused the characterization of map performances across different steps. Then testing map with different initial condition of fixations, just to make sure of the versatility of this technique. And then assess the ability of map to spatially profile synaptic proteome quantitatively by reproducing partially results that has been shown before using electron microscopy. So we focused on the coronal sections of the mouse brain, and especially in the subregion CA1 of the hippocampus. And I'm going to talk about two main combinations of antibodies for my first two aims. So we labeled ELX, the synaptic, so the active zone marker ELX, the voltage-gated calcium channel CAV 2.1, and all of the MAGOC protein from the postsynaptic density in one combination. And in another antibody combination, we labeled PSD95 and the essential subunit GLUN1 of an MGA receptor, as well as all of the AMPA receptor, all of the subunit of the AMPA receptors. So we label we use these two combination of antibody, as all of these proteins are challenging to visualize with conventional immunohistochemistry. So to begin with, let's take a look at the improvement in synaptic protein visualization. And so first, in the mouse CN1, I labeled for all of the antibody that I talked to you before. And what we can observe here is we know that in this subregion of the hippocampus, the excitatory synapses should be focused mainly in the stratum radiatum and not in the pyramidal layer. However, what we can see here for PanMaGOC, which is an excitatory marker, is that there is a homogeneous distribution throughout the stratum radiatum as well as the pyramidal layer. While ELX also present labeling where the nucleus is, which should not be the case, and the pattern of distribution of CAV 2.1 is not better for regular IHC, immunohistochemistry. Once we do the same link or the same labeling on tissue processed with the magnified analysis of the proteome, we can see a striking difference. So first, we can see that the signal to background ratio seems higher, and we obtain the right pattern of distribution for PanMaGOC with a marked difference between the stratum radiatum here and the pyramidal layer. If we take a closer look at the individual punctal level, we can see that only for map processed tissue, we have the presynaptic marker ELX and the postsynaptic PanMaGOC that are aligning one in front of another with a very good signal to background ratio, with sometimes calcium channel lying between PanMaGOC and ELX. Then we try to quantify this amount of colocalization. So to do so, we looked at the amount of colocalization between all of the puncta, so Palmaguk with ULX or Palmaguk with calcium channel or calcium channel with ULX. And we can see that only with map processed tissue presented a high rate of colocalization between every synaptic clusters. As control for the random colocalization, we flipped 2 channels out of the 3 channel for 20 gen to randomize the distribution. And so we observed a significant reduction of the amount of colocalization for map processed tissue only, suggesting that the amount of colocalization we have seen here for the regular IHC might be due to only random colocalization. Then we performed the same analysis for the other combination of antibody for PSG95, GLUN1, and GLUA2, and we observed the same pattern with a high rate of colocalization for map processed tissue, while the IHC procedure might be only give random colocalization. Then we tested this procedure on many different types of antibodies, obtained good staining for more than 40 proteins, mainly synaptic proteins, but also other type of proteins. Here we can see various synaptic proteins that can be labeled using MAP, with most of them having a very low background, and so a sharp difference between the background and the true signal. And as we can see, most of the time we have the puncta that are aligning for either a pre or a postsynaptic density protein, or for different postsynaptic proteins. Then we wanted to check which step of the procedure contributes the most to this gain of synaptic protein visualization. We reduced the possibility to 2 steps, the denaturation/clearing step, or the physical expansion of the tissue. So to test this, after labeling map processed tissue, I imaged them non-expanded, meaning that instead of putting the tissue in diluted PBS or in water, I kept them as regular 1x PBS to keep them limited expanded, so 1.4x expanded. And we can see that when we kept them not that expanded and we image them with a regular confocal microscope, we can still visualize the protein at the individual synapse level. So we can see here Palmaguk in red aligning with ULX and with calcium channel localized between these ULX and Palmaguk. So we can already see that even without having them fully expanded, we have again of synaptic protein visualization. Then the same tissue was expanded in water and imaged with the same imaging settings as for the 1.4x expanded. And we can observe that barely any signal can be visualized, mostly due to the dilution of the signal. So this is a parameter to take in account, that these techniques dilute a lot the fluorescence signal of the labeling that you may use. And once the imaging setting has been optimized, a more detailed view of the labeling distribution can be seen, allowing to even see maybe some nanoclusters, especially for ULX, aligning in front of Palmaguk with distinct puncta aligning in front of one Palmaguk. So here, the improvement of synaptic protein visualization is primarily due to the denaturation/clearing step, while the physical expansion of the tissue brings more detail to the visualization of the distribution of these proteins. I also realized the same with the same procedure with the other antibody combination, obtaining exactly the same results. Then we assessed the power of combining MAP with different microscopes. So first, we used a regular confocal microscope, if you know the LSM 700 from Zeiss, and we imaged only PSG95 to show we just imaged here PSG95. And what we can see here is already with a regular confocal microscope, we can locate the signal at the individual synapse level, at the individual puncta level. Then we use two super resolution microscopes, the LSM 880 and 980, which are processing super resolution microscope. It's not really a physical super resolution microscopes, which gives a better signal to background ratio. But also for the LSM 980, we can start seeing some higher intensity within one cluster, which may be the different nanocluster within the synapse. And then with the STED, which is a true super resolution microscope, we can clearly see the different nanocluster within one synapse for PSD 95, showing the capability of combining these super resolution microscopes with MAP, depending the amount of detail you may need for your experiments. And then we quantified the actual resolution that can be achieved using these different microscopes compared with expansion, the magnified analysis of the proteome. We've here the physical scale, so the actual scale, the actual resolution that you are observing below the microscope, and the biological scale when you take in account the expansion factor. And you can see with regular confocal microscopes, you can already reach a resolution between 120 nanometer and 60 nanometer. With this more processing super resolution microscope, you can reach up to 25 nanometer at the biological scale. And with the true super resolution microscopes, you can go all the way to 15 nanometer of resolution at the biological scale. So now for the next focus, we tested the different fixation conditions for the tissue to check the versatility of this technique. So first we used the original MAP procedure, which was using a mix of acrylamide with PFA to fix the brain. Then we fixed the tissue with PFA at 4%, which became our standard now. And then we freshly extracted the brain, the mouse brain, such as in an electrophysiology procedure. And then we incubated it in the MAP fixative. Or we do the same fresh extraction, and we incubate the tissue in formalin, which is the fixative used to fix human brain tissue for 1 week, 1 month, or after a couple of hours at 4 degrees in the fridge to mimic the postmortem condition that we can apply it on human brain. And so we can first observe that in all of these conditions, the signals show that a very good signal to background ratio with individual puncta that are aligning in front of another for Panmaguk and ULX. Then when we look at the quantification, we can see that for the signal to background ratio, this ratio is on average around 10 for all of the conditions and all of the antigens looking at. And for the coefficient of variation of the signal, even though there is significant difference between the different conditions, it is on the same order of magnitude, which is quite low compared to the intensity. So this shows that this technique is very versatile and can be applied on tissue fixed in many conditions. So which means that one brain can be used for different experiments if you want to combine the expansion microscopy with electrophysiology, or if you want to use it to use your tissue for different procedures such as transcriptomic or other type of analysis. So we even tried formalin fixed paraffin embedded mouse brain tissue, which has the closest conditions to the human brain preservation, which gave great staining quality also with individual punctal visualization of the synaptic proteins. So lastly, we assessed the ability of MAP to spatially profile the synaptic proteome quantitatively. And we looked at the differential distribution of AMPA and NMDA glutamatergic receptors, as well as the Shanks proteins between synapses made on interneurons and on pyramidal neurons in the hippocampus. So to do so, we use a transgenic mouse line expressing channel rhodopsin tagged to YFP in all hippocampal interneurons. And so we could look at the dendrites of these neurons expressing channel rhodopsin. And by having these dendrites, And using PenMaghook to locate all of the excitatory synapses, we could look at synapses made on these dendrites, so on interneurons, versus synapses made not on these dendrites, so on pyramidal neurons, which are the other input in the hippocampus. And then we co-stain either AMPA receptors or the essential subunit gluN1 of the NMDA receptors, and we could do the analysis and see the differential distribution of these glutamatergic receptors at these two types of synapses. And so what? Sorry, I forgot about this. So to do the quantification here, we use the technique called, we use the software called Arivis, which allow to generate a surface that model the fluorescence signal. And then we can do this analysis in 3D to be able to look at the distribution of these proteins in 3D and their intensity in the volume instead of doing an analysis in 2D. And to take in account the variability that there is between the experiments, we normalized this. We normalized the value that we obtain either for the volume or for fluorescence intensity for each individual cluster. So to do so, we took the value for each individual cluster and each parameter we looked at, whether it was the volume, the sum intensity, or the mean intensity. And we divide this individual value by the average obtained for the entire stack and all of the animals combined all together to try to take in account the variability between batch, but also the technical variability, which can be quite high using expansion microscopy or magnified analysis of the proteome. And so once this is taken into account for the quantification, we observed that for PenMaghook, the volume is significantly higher on synapses made on interneurons, showing bigger synapses made on interneuron than in pyramidal neurons, while the mean intensity, which can be related to the density of proteins within these synapses, is the same between synapses made on interneuron or pyramidal neurons. For AMPA receptor, both the mean and sum intensities of synaptic clusters are significantly higher on interneurons, while the opposite trend was found for NMDA receptors. All of these results were expected based on electron microscopy data, showing the ability of MAP to be quantitative for the study of synapses. Also, we wanted to push a bit further, looking at Schenk's protein, because a graduate student in our lab who was working on the cerebellum found some interesting differential distribution in Schenk protein at excitatory synapses. So we wanted to do the same in the hippocampus, knowing that all of the proteins of this family of proteins are involved in psychiatric disorders. So it may be interesting to see how the distribution might be different between different types of synapses. And what we observed is only Schenk2 present an actual differential distribution with a higher mean and sum intensity on synapses made on pyramidal neurons versus on interneurons. While for Schenk1 and Schenk3, even though there is a small significant difference with higher value for synapses made on interneurons, this could be explained with the higher volume of synapses made on interneurons versus pyramidal neurons, showing that maybe only Schenk2 would present a differential distribution with a lack of Schenk2 in synapses made on interneurons. And to our knowledge, such difference have not been reported prior to this study, showing the ability of this procedure to unveil a new pattern of distribution for synaptic proteins. So in conclusion for this part, we saw that this technique allows a gain of antigen visualization, primarily due to a gain of antigen accessibility. MAP can be performed on tissue fixed with various initial fixation conditions, showing its great versatility. And also it's able to spatially profile the synaptic proteome quantitatively. So just to speak a bit about what's next for me, I'm going to study the synaptic pathology of epilepsy in human tissue. So I will use MAP to study the synaptic pathology of human hippocampus in the context of major temporal lobe epilepsy. And here we can see in this partially expanded human hippocampus, the scale which I need to look at. So it's on the order, here's on 2 millimeters. So if we look here, it would be around 10 millimeters by maybe 8 or 10 millimeters. And if I want to look at all of the subregion of the hippocampus in a significant amount, I will need to image a lot of signal at once. That's why we acquired the latest light sheet microscope and the one from Zeiss. I'm going to come back to it later about some struggle we've been having with this microscope. But first, I can show you some initial labeling that we obtained using MAP on human tissue. And we can see by labeling PenMaguk and then the two different subunits GluA2 and GluA1, we obtain a very good signal with a very good signal to background ratio with this labeling colocalizing with one another, showing even more the versatility and the application of this technique to directly human brain tissue. And this comes from biopsies, so bank of tissue that are stored for months or year in paraffin block, which means that this technique could potentially unveil a whole new study, a whole new world of study, using all of these bank of tissue that are conserved for months or year, which when you do regularly IHC on it, it doesn't give a good signal or a lot of autofluorescence. While here, using this procedure, you get rid of most of the autofluorescence and you still keep a very good antigenicity of the protein of interest to label them. So I was telling you about, but I'm using the latest light sheet microscopy. And for the ones who don't know about this technique, these combine the concept of the light sheet microscopy, so illuminating planes of your tissue with a sheet of light, which is gentler on your tissue, but also faster to do the scanning, with a specific objective that allows to keep a very high resolution. So we are around we are the order of 300 nanometer of resolution using this technique, while being 500 times faster than regular confocal microscopy. However, because this technology is quite new, there is a bit of troubleshooting using this technique. And so one of the first things that we noticed was the processing time. So the images generated with this technique might be very big. So right now, me, with the amount of tissue that I need to acquire, I routinely generate several hundred gigabytes of data with just one imaging session to maybe a terabyte of data at once. And so these data need to be processed because here, due to the way we acquire the data, the plane of view is skewed. And so you need to deskew this plane of view. And then you need to deconvolve the data to get a good signal to background ratio. And this takes a lot of time, especially on a very big file of hundreds of gigabytes or terabytes of data. So we tried a couple of things. There is this in Zen, they have their own latest light sheet processing. And when we use with the most optimal settings for one 40 gigabyte compressed file, so the actual file is bigger, around 100 or 160 gigabytes, it takes up to 17 hours to be processed. You can play around and not do the optimized way to do the processing using Zen. And instead of correcting for the background, you can subtract the background after the processing, and you can reduce the time of processing to 4 hours, which is still quite high, knowing that now I will need to process hundreds of gigabytes. So we are working on the side with a graduate student to have a processing done on Python instead of having it directly in Zen, as Zen might not be the most efficient way to process such data. Knowing that, as I was saying, the amount of data that I can acquire can be quite big. If I would like to do the entire human hippocampus, I would generate more than 800 terabytes of data, which is really big and cannot be handled using the Zen process. Another issue that we've been running into, but which might be resolved now, is a loss of focus. So our main guess when we are doing the imaging, the objective are not moving, but the stage is moving. And as I was telling you, on expanded tissue, you are moving by millimeters during your scanning. And maybe during this movement on the order of millimeters, the stage lose the focus, but we are not talking about a big loss of focus. We're talking about a few micrometer, but in my case, this few micrometer is enough to completely lose the focus at some point when we do this millimeter scanning. But recently they updated the Zen software with a new autofocus mode. So it's called reflex autofocus. I just tested it last week, and it allows, I think, it allows to, when you set a certain focus, it allows to calculate the distance between this focus and the reflection of your cover slip. And using this distance, it makes sure that at every tile that you are taking, it keeps the same distances between this focus and the reflection of the cover slip, which was quite helpful. I tried it recently, last week and this week, and it allows for big field of view. So I didn't do such a big field of view, but maybe 2 millimeters by 2 millimeters. I was able to keep quite constant the focus using this new autofocus reflex from the Zen software. And then I'm not sure, I tried to play this a couple of times, but no, it's not working. What I wanted also to show you here was that when you image using the lattice light sheet, because of the physics of a sheet of light, you will have a very thin sheet of light in the center of the sheet of light, but on the top you will have this cone of light. So you will have a convergence of the light only for a certain part, and this cone generates a lower signal. So what you should have seen on these images is like on the top and the bottom of your stack of acquisition, you might have lower signal, and in the center, you would have a higher amount of signal. So I quantified it by integrating this signal for every stack. For every plane, every stack position, I look at the integrated intensity for the 2 dimension of light sheet that gives the better precision. So there is a very thin sheet of light and a medium sheet of light. And you can see, especially for the thinnest sheet of light, that you have a heterogeneous distribution of the integrated intensity with lower values on the edges, so on the top and the bottom of your acquisition, and Bell distribution of the integrated intensity for the center, where the signal is stronger. For the medium sheet of light, you have this sort of plateau in the center, but you are still using up to 50% of your field of acquisition. So this is also something else to take in account. We had our engineer from Zeiss coming to do the maintenance, so I need to redo this analysis to see if with the maintenance we get less variability in the distribution of the signal using the lattice light sheet. But still, this is something that you may need to consider, because I know some of you uses these lattice light sheets, so it might also be something to consider when using this technique, and especially if you think about using the lattice light sheet in combination with expansion microscopy. So on this, I would like to thank my two labs, so Mark Cembrowski lab and Marie's lab, for their support and their help. And a big thank also to Jeffrey Ledoux as well as Peng Zhang, who is now a PI in Case Western University, who helped me a lot with this procedure, either it was for the imaging or the processing of the images, or just to implement this procedure at the very beginning. And so I would be happy to take any questions if anyone has.
All right, thank you, Mathias, so much for that. A wonderful presentation and all the tips and tricks and your experiences with the troubleshooting. I'm gonna wait and see if our audience has any questions. Yeah, you're welcome to use the chat and then we can read your questions out loud. We'll give that a minute or two.
I had maybe more of a comment. We were also having issues with focal drift with live imaging. And we found leaving the instrument on all the time helps. So we don't turn it off in between like day to day. We just leave it on all the time.
Yes, so that's something that when the engineer came last week, he told us now the new advices from Zeiss, indication from Zeiss to use the latest light sheet is to let the microscope on at least 8 hours before using it. And so for us, it means, because I'm doing acquisition almost every day, it means just letting it on all the time.
Yeah, I think that's been our approach as well. And thank you for that. It was very comprehensive. Nice to see the biological examples as well as some of the technical tips. Excellent. Thank you.
Thank you very much.
Have you tried doing anything with the large data sets in terms of also looking at like proximity between synaptic clusters and really mapping out distances and things like that? Or have you focused mostly on the quantification of each individual spot?
So me personally, for this paper, we just look at the quantification of individual spots, but I know that so Peng, who is here, Peng is using the expansion microscopy also now for his studies. Oh, where's my mouse? Okay. And so he's looking at the distance between the pre and the post synaptic density, because he's looking at this mouse line that can be related with autism. And so he's looking if some of the mutations that he's looking at may alter the distance between the active zone and the post synaptic density proteins. The thing is, so with fresh tissue, especially with mouse tissue, it might be something that you can do, but with FFPE tissue, so the tissue I'm looking at now, maybe if we go back to it, one of the issue with it, I think it's because it was fixed for such a big amount of time. You have too much cross-linking between the proteins, and especially with proteins that are very close to one another. So in this case, for Penmagook and ULX, you can see that there is not much distance between ULX and Penmagook. You can see that they are not completely overlapping, but they are still overlapping partially. While when I look at fresh tissue, for example, the one just before, you have a better separation between Penmagook and ULX. I assume it's due to the chemistry. So when you fix with formalin and you keep it in this paraffin block, first in formalin, usually they keep it in formalin for quite a while before embedding it in the paraffin. And then they stay in these paraffin blocks for months or year. And so when I do the procedure on this tissue, I assume that there is too much cross-linking between the pre and the post-synaptic density. And so even though you do the expansion and you tear them a bit apart, I think they are too cross-linked to allow the gel to really pull them apart. So I'm not sure if it would be a fair comparison to look at the distance between these components in this very specific case of formalin-fixed paraffin-embedded tissue. If you just look at mouse brain tissue and you look at fresh mouse brain tissue, in that case, yes, I think you may be able to look at the distance between synaptic components.
Yeah, that's a really good point. It actually leads a little bit into my last comment is, I don't know if you're aware, but the Douglas Hospital in Montreal has the world's biggest brain bank of mental health, from mental health disorders. And they have it linked to all of the patient history, you know, and anonymized and so on. So I can send you an email with a link to that. But that tissue would be like you're describing. I would assume it's a tissue bank. So I would imagine they get new tissue on a regular basis. But I would think a lot of it has been kept under the conditions you're describing. But it may be of interest to looking for localization rather than you know, specifically the distances.
Yes, no, yes, definitely. I think it's yeah, it's very good comment. So that's what I was trying to tell at the end is like, so even though, for example, you cannot look at distances within the synapses, it still gives a way better antigenicity of the proteins of interest. So especially for human brain tissue. So for all of these tissue that are now in biobanks and may not be used for specific proteins because usually with regular IHE, you don't have very good signal. You can still use these, just even if it's just to look at your protein of interest. Oh yeah, I will look into this definitely. So I had a few questions in the chat. So for what was the software I used? The software I used is a Revis. So it's a software that now belongs to Zeiss. It's this software is I don't know, maybe you know Imaris, which was popular before Revis. It's a bit the same concept that Imaris. So you do a 3D rendering of your signal, and then you generate this surface. The thing with Revis versus Imaris is at the time where I was using the two software, you had more options with a Revis on the parameters you can look at, as well as you can already do some prior analysis within a Revis, that while with Imaris you couldn't do it. And also at that time, I don't know right now, a Revis could handle better very big tissue, very large field of view, while Imaris was better suited for smaller field of view. And for example, we have people using a Revis for the entire reconstruction of a mouse brain using the light sheet, not the latest light sheet. And so having this software that can handle very big data set was very helpful in our case, and also in my case, because I'm using very big data. I'm generating very big data. Oh, thank you very much, George. Yeah, about the across microscope and spatial frequency. And I have to say for this, a big shout out to Jeff Ledoux. I did the acquisition, but he helped me with the analysis across the different microscope, and he was the one strongly suggesting to do this analysis. So big shout out to Jeff for this specific. Oh, so the nanocolumn alignment. So that's a great question. We just had a Canadian symposium on the synapse 2 weeks ago, and I was talking with Dr. Thomas Blampier. So I mean, if you know about nanocolumn alignment, you may know about Thomas Blampier. And I was asking him his opinion about using the expansion microscopy to look at nanocolumn alignment. So you may be aware of a paper that he published in collaboration with Edward Boyden in 2022, where they used the expansion microscopy to look at this nanocolumn alignment. He said at the end, Thomas Blampied didn't perform the technique. It was Edward Boyden that did most of the technique to look at this nanocolumn alignment, because the protocol at that time was still a bit in development. So it was better to have Edward Boyden's lab performing the technique than him. But he told me that he is not fully confident using expansion microscopy or MAP to look at the nanocolumn alignment, for the same reason I was talking about FFPE tissue, because the tissue are so close together. So maybe we can look a bit before. Yeah, so here, because the tissue the proteins are so close within the nanocolumn, when you do the initial fixation step, so PFA fixation or whatever type of fixation you would do, you may already cross-link them too much. And because these nanocolumns are very close together, but also a bit dynamic, by fixing them and having them cross-linked together, sometimes when you do the expansion microscopy, they are not they are not distributed the way you should expect them to distribute, and having like very big clumps of intensity, while the nanocolumn should be a bit thinner. So he told me ultimately using MAP or expansion microscopy may not be the best to look at nanocolumn on fixed tissue. He is right now using, I don't know if you checked, he has a very recent paper he published in Journal of Neuroscience. He used DNA paint, instead of expansion microscopy to look at nanocolumn alignment in fixed tissue. So that might be something to investigate, or it would really require a lot of verifications and maybe fine-tune adjustment to the techniques, just to make sure that you keep the expected distribution of the nanocolumn of the nanocolumn using this technique. Did you have to use yes, so for the GFP, yes, I had to use GFP antibody for the post expansion microscopy labeling. However, now we've been trying different denaturation conditions, and we noticed that you can do gentler denaturation condition. The initial denaturation condition were 37 degrees for 2 hours, and then 95 degrees for 1 hour. 95 degrees for 1 hour is very harsh on the GFP. You would just get rid of the fluorescent signal. Now we use also 65 degrees overnight, which is not as harsh on the tissue. And using these longer denaturation but gentler condition might keep the fluorescence of GFP. So if you have intrinsic fluorescence within your tissue, for example, GFP, TD tomato, maybe by having gentler conditions of denaturation but longer, you might be able to keep the fluorescence of your fluorescent of your fluorophore. Can it work with non-Zeiss dataset? You mean, oh yes, the reverse? Yeah. It could work. You just need to make sure that you do all of the processing beforehand. So if you do, for example, every scan or other type of lattice light sheet, the 3i, I think it's a 3i, or the in-house lattice light sheet, as long as your signal is already processed, deconvolution, maybe deskewing, or any type of processing, then you can just put whatever signal, whatever type of file in a revis. It's going to create this SIS file out of your initial file of interest. So you are not going to do any modification on your file. So either it would be CZI or the leaf file from Leica or any other type of microscope you would have. It would not do any modification on this file. It would create kind of a clone SIS file, and then it would do the modification on this file. So I think a revis is compatible with most of the microscopy file, whether it's from Leica, Olympus, Nikon, Zeiss. What is the total size in depth of your lattice light sheet acquisition? So in my specific case, I'm just looking at one plan of acquisition, which is, if I'm correct, 30 micrometer of depth. But we've pushed a bit when we tried the latest light sheet initially. We tried to push it as much as possible, and we were able to acquire several hundred micrometers, micrometers of depth in I think it was in a 300 or 400 micrometer thick tissue that was expanded. We were able to acquire in depth up to, I think it was 300 or 400 micrometer at least, and we haven't pushed as far as possible. Looking at the latest light sheet microscope, the way it works is you have a certain magnitude, you can move the focus, and it goes from minus, if I'm correct, it goes from minus 1000 microns to plus 1000 micron. So depending where your sample sits, you may have all of these magnitude to play with. But the same, your tissue will not sit, for example, at minus 1000, and you can go for 2000 micrometer. Usually in my specific case, the tissue sits around minus 500 micron, and then I can play between minus 500 micron to minus 1000 micron. So I have 500 micron I can play with if I would have this amount of tissue I would like to acquire. So yeah, this technique can go a bit in depth, but the same. You need to consider that this a microscope was designed originally for live cell imaging, so in cell culture, and so it will not do any depth as for example a light sheet microscope. Even if it has the light sheet technology, it will not go too much in depth, so you would be more limited than with a light sheet microscope, definitely. No worries. Did I miss any questions here? No.
All right, so thank you so much, Matthias, for answering all of the questions, and so detailed. I think we all really appreciate that. If there's no more questions, is there something else in the chat? Okay. Yeah, so thank you so much, and we'll be posting the recording onto our YouTube channel. And I guess if there's anyone else who has questions, could they contact you by any chance by
yes,
email?
definitely. Yes.
Okay, perfect. All right, thank you, everybody.
Thank you.
Thank you very much for the invitation
the
again.
next meeting, Natalie, before
Oh, yeah.
everybody leaves.
Yep.
Thank you
We're
so
gonna
much.
take a break for the summer, right? And meet again in September. Was that the plan?
Yeah, I think so next month, so July, we're still planning with the Australian counterparts to see if we can secure the time and date for the Australian hosted meeting. And then for August, we'll be taking a break. And then we'll see if Australia wants to continue in September. If not, then we will resume our meetings in October. Okay, all right.
Thank you.